Okay, so welcome everybody. I'm glad that you are all here with these changing weather conditions, so thank you for coming. And today we have Marta Garrigós. She's a PhD student here at EBD. Her supervisors are Josué Martínez de la Puente and María José Ruiz López. And she's going to present a PhD plan of her thesis. So I remind you that normally PhD talks are 20 minutes of talk and 10 minutes of questions, but we only have one PhD student, we have plenty of time, so if you would like to do more questions or if you would like to take a little bit more time, you have it. So I hope you enjoy the talk, and the floor is yours, Marta.
Okay, thank you for the introduction. Hello everyone. Today I'm going to present you my thesis, which is focused on the interactions between mosquitoes, their microbiota, and avian pathogens. First, I would like to give you a brief introduction of myself and my background. I studied biology at the University of Seville. Then I studied 2 master degrees, one of them in Biodiversity and Conservation Biology at the Pablo de Olavide University, and the other one in Bioinformatic and Biostatistics at the Open University of Catalonia. During my last master, I got a JAE-Intro grant here at the EBD. And finally, I started my PhD with a FPI contract at the University of Granada in 2022. But I recently moved here to the EBD, and hopefully I will finish my PhD in 2026. Vector-borne diseases cause more than 700,000 of human deaths annually, and they are produced by pathogens that need an organism, the vector, to be transmitted from one host to another. And many of these vectors are arthropods, including ticks, fleas, biting midges, and our main character today, mosquitoes. Mosquitoes are indeed the deadliest animals in the world due to the pathogen they transmit, especially for the malaria, which is a blood parasite of the genus Plasmodium that causes around 500,000 deaths every year. But mosquitoes also transmit pathogens to other vertebrates, and among these pathogens we find a wide variety of organisms, including filarial worms, protozoans, such as those of the genus Plasmodium, that cause malaria not only to humans but also to other vertebrates, and many different viruses, such as the West Nile virus. In my thesis, we focused in 2 pathogens that naturally circulate in southern Spain, which are West Nile virus and avian Plasmodium, and that present similar transmission dynamics. They both are transmitted from mosquitoes to birds, which are their only reservoirs. But they also present some differences. Unless avian Plasmodium that only affect birds, West Nile virus can occasionally affect other vertebrates, such as horses or humans, but they act as dead-end hosts, meaning that they cannot transmit back this virus to the mosquito. They also share vectors that are mosquitoes of the genus Culex, and here in southern Spain they are mainly transmitted by Culex pipiens and Culex perexiguus. But the relevance of these 2 vectors in transmitting these pathogens is a bit different. While Culex pipiens is considered the main vector of avian Plasmodium, Culex perexiguus is considered the main vector of West Nile virus here in southern Spain. This is important. There are numerous factors that affect the transmission of pathogens by the mosquitoes, and these factors can be environmental, such as the humidity, the temperature, or the food availability, but also intrinsic, including the age, the sex of the mosquito, and also the presence of the symbiotic bacteria that are part of the mosquito microbiota. This microbiota is acquired by the mosquito during the larval stage from the breeding sites, and therefore it is highly dependent on the environment. Then, once the mosquito emerges, it loses most part of this microbiota, and the remaining microbiota composition will be modified by their diet, which is, in the case of both females and males, the sugar of the plant in which they feed, and only in the case of females, the blood meals they take from the vertebrates, which will modify this microbiota composition at least during a short period of time. So this mosquito microbiota affect all the aspects that are involved in the vectorial capacity, which is an epidemiological term that informs about the relevance of a given mosquito population in transmitting one pathogen. These factors that affect the vectorial capacity are, for example, the vector density, the vector beating rate, or the pathogen extrinsic incubation period, but also the mosquito daily survival and the vector competence, which are the aspect in which I focus here in my thesis. The vector competence gives us an idea of how well the pathogen is developing inside the specific vector, and therefore how well it will be transmitted. And this vector competence is a consequence of a number of different mechanisms that happen between the vector and the pathogen, and the mosquito microbiota can affect many of these mechanisms, including direct mechanisms such as releasing antimicrobial molecules, producing oxidative stress, but also can affect this vector competence indirectly by stimulating the immune response of the vector. So based on this, in my thesis, our main aim is to unravel the interactions between mosquitoes, their microbiota, and avian pathogens, and it can be divided in 3 main sections and 9 chapters. Section 1 presents only 1 chapter, and here we aim to characterize the microbiota of mosquitoes from southern Spain and to explore the associations between this microbiota and different variables. In section 2, we focus in the interactions between the mosquito microbiota and avian pathogens, and we studied West Nile virus, avian malaria, and using different approaches. And in section 3, we focus in the vector, and we want to explore the response of the vector against both the exposure to pathogens and also by the disruption of their microbiota. To make it simple, I'm going to explain you a bit the general methods that are used in more than 1 chapter. I'll start with the sample collection, which have been conducted in Andalusia, in the provinces of Málaga, Granada, or Seville. And in the case of correlational studies, we sample the adults directly using these 2 types of traps. Here we have a CDC UV light trap, and here we have a BG sentinel trap. And in the case of experimental studies, we collect the larvae of the mosquito, bring them to the laboratory, and let them emerge in climatic chambers under controlled conditions. And in the meanwhile, we expose them to different treatments. For example, we expose them to antibiotics or to parasites. Then, when we have the samples, the adult mosquitoes, when we collect them from the field, we test it for the presence of parasites using PCR in the case of avian malaria and qPCR in the case of West Nile virus. And then we look at the mosquito microbiota or at the mosquito gene expression, or both of them, depending on the chapter. When we look at the mosquito microbiota, we extract the DNA of the sample, then we sequence the 16S ribosomal RNA gene, which is a bacterial gene used for metabolic coding techniques. And then we use different bioinformatics software to process these sequences, for example, TEMP2. Then we perform the statistical analysis in R using different packages of the bioconductor environment. When we look at the gene expression of the mosquitoes, we extract and sequence the RNA of these mosquitoes, process the sequences using, again, different bioinformatics software such as STAR or RSEM. And again, we use R and bioconductor packages to perform the statistical analysis. So now I'm going to go through the different chapters in more detail. I'll start with section 1, which has only one chapter, in which we assess the effect of space, time, and species in mosquito microbiota. Here, we conducted an extensive sampling consisting in 10 sampling sessions from May to November of 2022 in 5 localities of southern Spain, of the provinces of Granada and Malaga. And we collected mosquitoes of 3 different species: Culex pipiens, Aedes albopictus, which is the invasive tiger mosquito, and Culiceta longiarelata. And we are now analyzing this microbiota. Our hypothesis here is that mosquito microbiota may vary among species, but also among localities and seasons, as remember that the environment has a strong effect in the mosquito microbiota. Then we go to section 2, in which we focus on the interactions between avian pathogens and mosquito microbiota. And we start with the West Nile virus. And we first, in chapter 2, conducted a literature review on the interactions between West Nile virus and the microbiota of Culex pipiens vectors. We found that only 7 studies focused in this topic, and among them 5 studied exclusively the Wolbachia bacteria, which is a really studied genus of bacteria. And we found that in experimental studies, the relative abundance of Wolbachia bacteria was negatively correlated with the West Nile virus, while the relative abundance of other bacteria, such as the Ratian Enterobacter and the Biodiversity, were positively correlated with the virus. However, in the case of correlational studies, they were really scarce, and also the results were contradictory, so we cannot draw any conclusion about it. So the next step here was to assess this interaction between West Nile virus and the mosquito microbiota, but using field collected mosquitoes and in our system. So we collected Culex perexiguus females from La Dehesa de Abajo, which is a natural area here in Seville, in which a big West Nile virus outbreak occurred in 2020. And our objectives here was to identify the Culex perexiguus microbiota and to compare the microbiota composition between West Nile virus positive and West Nile virus negative pools. What we observed here is first that the microbiota of these mosquitoes was dominated by the families Burkholdeриaceae and Ewinaceae. And also we didn't find any significant difference in the microbiota composition or in the biodiversity between positive and negative pools. Next, the rest of the chapters in this section will focus on avian malaria parasites. We first characterized the avian plasmodium lineages that are circulating in mosquitoes from southern Spain. So, using the mosquitoes collected in 2022, we tested for the presence of avian plasmodium DNA, and we found that only in Culex pipiens pools there was plasmodium DNA, specifically in 31 out of 165 pools. And we identified a total of 8 different lineages that were differently distributed in the 5 localities we sampled. The next step here is to identify the microbiota composition of these Culex pipiens mosquitoes and to explore the potential associations between this microbiota and the infection by avian plasmodium. And our hypothesis here is that avian plasmodium infection may be correlated with mosquito microbiota diversity and composition, and we expect the strength and the sign of this correlation to be conditioned by the microbiota taxa and also by the parasite lineage. So then, to finish this section, we wanted to assess this interaction between mosquito microbiota and avian plasmodium, but using an experimental approach. So we allowed Culex pipiens females to feed on one house sparrow that was infected by Plasmodium relictum and on another house sparrow that was uninfected. And we obtained from those mosquitoes that fed on the infected bird, we have infected and uninfected mosquitoes. And here we expected that mosquitoes exposed to avian Plasmodium may modify their microbiota composition, and also that infected mosquitoes may show an increased bacterial diversity based on the current literature. So our preliminary results are here. Here I show you the relative abundance of the most abundant genera in these mosquitoes. And we can see first that Wolbachia dominated the microbiome of these mosquitoes that were collected in Seville. And we can also see that infected mosquitoes seem to show a slightly higher diversity and also a slightly higher relative abundance of other taxa in comparison to the other groups. For example, this genus, Faeculibacterium, seems to be in a slightly higher relative abundance. So finally, we go to section 3, in which we focus on the vector response. And we start by studying the transcriptional response of Culex pipiens females infected by 2 different avian malaria species. So here we allowed again Culex pipiens females to feed on one house sparrow that was infected by Plasmodium relictum, another house sparrow infected by Plasmodium catemerium, and on an uninfected house sparrow. And at 3 different time points, 1 day, 10 days, and 21 days post-infection, we studied the gene expression and compared the gene expression of these 3 experimental groups pairwise. The first thing we observed is that the most transcriptomic response of the mosquitoes was at 1 day post-infection, which is where I'm showing you here in this bar plot, that represent the total number of differentially expressed genes in the different comparisons. We can see that here the comparison that showed the highest number of differentially expressed genes was mosquitoes infected by Plasmodium relictum versus those infected by Plasmodium catemerium, which means that the response against the parasite by the mosquitoes is quite different depending on the parasite which is infecting it. Then the next comparison showing a higher number of differentially expressed genes is mosquitoes infected by Plasmodium relictum versus controls, and then mosquitoes infected by Plasmodium catemerium versus controls. So it seems that the response is higher when the parasite that is infecting the mosquito is Plasmodium relictum. But in both cases, we find several genes that are related to the immune response of the mosquito. For example, these 2, secropin and spatzel, are involved in the toll pathway, and these 2 are also involved in the response against pathogens. So then the next step is to assess the response of the mosquitoes, but against a disruption in their microbiota. In chapter 8, we assess how this disruption in the microbiota affect the mosquito survival, which, remember, is involved in the vectorial capacity. And we combine an antibiotic treatment during larval and adult stages and obtain 4 different experimental groups. Mosquitoes that were treated with antibiotics during both larval and adult stages, mosquitoes treated during the larval stage, mosquitoes treated during the adult stage, and untreated mosquitoes, which I'm going to call controls. So we then, during 68 days, assess the survival rate of 2,723 mosquitoes, females and males. The main result here was that only for females, the antibiotic treatment during the larval stage increased the adult survival. In this plot, we see in the y-axis the female survival rate, and in the x-axis the day of experiment from day 1 to day 68. So here we can see that these two lines, the darker ones, correspond to mosquitoes treated during the larval stage and mosquitoes treated, mosquitoes not treated, sorry. So they showed significant differences. And finally, we wanted to know how this antibiotic exposure was exactly affecting the mosquito microbiota, and also how it affected the gene expression of the mosquitoes. So using mosquitoes from the same experiment, here you can see the microbiota composition of these mosquitoes, again the relative abundance of the most abundant genera. And we can see first that, again, Wolbachia dominated the microbiota of these mosquitoes that were collected in Granada, in another area, and also that the antibiotic seems to, of course, it is affecting the microbiota of the mosquito, and it seems to increase the biodiversity by decreasing the relative abundance of Wolbachia in favor of other taxa. Sorry. So, how does it affect mosquito gene expression? In this principal component plot, we can see several things. First, the principal component 1, which explains the 23% of the variance, seems to be explained mostly by the larval treatment. If we divide the plot into vertically, we can see that at the right of the plot, there are all these samples that correspond to mosquitoes that were treated with antibiotics during the larval stage, regardless the treatment during the adult stage. And on the left, we can see those mosquitoes that were not treated during the larval stage. And a similar thing happens with the principal component 2, but with the adult treatment. If we look at the top of the plot, there are all these samples, or almost all these samples, corresponding to mosquitoes that were treated during the adult stage. And here on the bottom, we have the samples corresponding to those mosquitoes that were not treated during the adult stage. If we look at the specific genes that are being triggered by the different treatments, here we have the total number of differentially expressed genes by each of the treatments. On the left, we have the treatment during both larval and adult stages. On the top, we have the treatment during the larval stage, and here on the right, the treatment during the adult stage. So we can see that expectantly, the treatment that triggered the higher differential expression was the treatment during larval and adult stages, followed by the treatment during the larval stage and the treatment during the adult stage, which match with the disruption we were making to the mosquitoes, to the mosquito microbiota, sorry. And among these genes, especially those upregulated by the treatment during both stages and during the larval stage, we found a lot of genes related to the immune response of the mosquitoes. Again, secropines involved in the TOD pathway, heat shock proteins, etc. We are now reaching the finish of the talk. I would like to highlight the collaboration we are making during my thesis. We have collaborated with many researchers from my old institution, from the University of Granada. Also, there is the institutions, the EBD, the IBIMA, international institutions such as the CONICET in Argentina. And also I would like to mention the stays I'm doing. I just came back from Sweden. I have been studying with Elin Bideva how they analyze the microbiota data. And the next stay will take place from January to April in 2025 with Mathilde Hendring at the Institut Pasteur of the French Guiana. I'm going to finish by showing you the progress we are making during my thesis. For now, 5 chapters are already published, 2 chapters are in preparation, the manuscripts, and I have just received the data of the other 2 chapters that I will analyze in the next months. So, thank you very much for your attention and for coming in such a bad day. and I will be happy to answer any question you have.
Thank you very much, Marta, for this impressive talk and this impressive outline. 9 chapters, wow. So we have plenty of time for questions.
So what are you going to do in your stay that you have planned? Was it next year?
Yeah, it is not included yet in the thesis, but my plan is to learn a new technique in which they remove totally the microbiota of the mosquitoes and then to expose them to different treatments. So this is what I'm going to do. I don't know if I will have time to include it in the thesis. I hope so. But for now, as the thesis is too long already, I decided to not include it now in this talk.
Well, thank you very much for the presentation. I think it was very nice. And when looking at presentations like yours and thinking about microbiota, is it possible to make predictions? I mean, you can predict, I was wondering, the work is very descriptive so far, no? Is it possible to predict what could be the changes? What would you expect if they are the mosquitoes that are sampled here and the mosquitoes sampled there? Can you changes? Is there any hypothesis that you can formulate, or so far we're still learning the meaning of the microbiome and just describing the patterns, and later on we'll try to interpret the patterns?
Yeah, it's quite complex, of course, the microbiome world, but for this, you mean in the field, it's really difficult to predict. I don't think you can do it, but this is the reason why I'm going to the UIAN, because my plan there is to remove the mosquitoes' microbiome and then to expose them to different bacteria in order to see if what type of factors are involved in the bacteria that are maintained by the mosquito as microbiome, if there are some evolutionary aspect or is just random. So it's quite difficult so far. It is a really new field, so this is why almost all the work done so far is the only thing you can do is to perform experiments, try to remove the mosquito microbiota and to expose them to different bacteria, or to repeat a lot of time the experiments and see if how deterministic it can be, the microbiota that the mosquito. I don't know if I'm answering your question, but it's really difficult to predict it.
Yeah, well, it's what I was wondering, no? It's what was the level of knowledge, because I assume that the microbiome will include both pathogens and symbionts and
a lot of different things, but you have to go more deeply, to know this. And we are just like about this topic, I don't know how to say it, but like step by step, you have to first look at what you have in the field and try to do simple experiments in the lab, and step by step learning small things, and maybe in a future we can do things that are more I don't know how to say it, can you help me, María? Like more...
No, it's great. It's very clear.
Basically what happens is that you could make predictions. For example, you can start making predictions for famous species like Anopheles or Aedes. The microbiome work is more developed and they have huge colonies. And that has been studied more, so they know more how it interacts with Plasmodium, how it interacts with other diseases. In our case, we have to start in the beginning. So she's doing like the groundwork. Like, what do we have, for example, in Culex perexigus? It is the first time, more or less, that it's been done, because most of the work has been done in Culex pipiens. So yeah, we are starting to build that framework, but she really is interested in, like, as she said, like moving and trying to understand how can you make predictions? And to do that, you need to remove the microbiome and do experiments. So she will move in that direction.
Thank you.
We also have 2 questions online, so I will proceed with those before we get back to the audience. Thank you very much for this interesting talk. I would like to ask, what analysis did you perform to find the correlation with Enterobacter you showed at the beginning of the talk? This is asked by Daniel Sanchez.
Enterobacteria?
Enterobacter.
Yeah, I think he's referring to literature review. So right now I don't remember exactly the analysis, but if I remember well, I would like to know if he means the bioinformatic analysis or the experiment, or so. I'm not sure.
He might get back to us.
Okay, yeah.
He also has a second question, so maybe I can proceed with this one. He also would like to ask whether you find higher or lower bacteria species richness in infectious mosquitoes microbiome.
Why? Sorry, I didn't understand. Why? He's asking why I expect that?
Whether you find higher or lower bacteria species richness in infected mosquito microbiomes.
Yeah, in the, sorry, I don't know if I'm understanding. He means, if that person means, here, we found a slightly higher diversity in infected mosquitoes. Yeah?
Yeah.
So is the question?
Yeah, that's the question.
Okay.
Thank you.
Yeah, thank you. So I agree with Carlos about the lack of the mechanisms underlying the patterns that you may expect here. But I agree, this is an emerging field, and now what we need to do is to describe exactly these kind of things. So I have a collaboration in the Netherlands on the microbiota in Arabidopsis, and what we are seeing is a core microbiota, and then each population with a different diversity of microorganisms inside the seed. Why? We don't know. The function of it, we don't know. Yeah. But now we have to describe this. But in any case, one way to move forward in this direction perhaps would be to identify the species, because you have genus and big groups. Are you planning to grow microorganisms in petri dishes to identify the species, for example?
Yeah, the thing is, using 16S RNA gene, it's really difficult to reach this, to know the species, because they are very conservative genes. So it's difficult to do that. I think the better thing to do that is to sequence the whole genome of all the bacteria, and of course we could do that, but it's really expensive. So maybe in the future, but right now we are using these techniques, and it is really difficult to go to the species. I mean, some of the sequence can be identified to a species, but it will be difficult to make comparisons, because if most of them, you only have the information about the genus, you cannot make statistical comparisons. So in a descriptive way, yes, maybe we can do it. We can describe at least what we have, what we know, the species level.
Yeah, but what I mean is to grow them on petri dishes using different media.
Okay,
that's what I mean.
Okay, to cultivate them.
Yeah.
I don't know. I don't think we are thinking about it, at least in right now, because in my thesis we have finished with the sample collection, with the experiments and this. But of course, in the future, it can be in the French Guiana. Of course, we will do it, because they have the necessary tools to do it.
Okay, and the other way perhaps to think about is to use mutagenesis on microorganisms, and see, the effects of different variants and these kind of things on the system. So trying to make a step forward, adding complexity and trying to get away from pure description. But I agree, it's the time to see what we have and everything.
Yeah, no, but of course, the next step is to perform the experiment, like looking at a specific taxa or specific genes in the case of transcriptomes, and but for now, we are starting in this field. But in the future, of course, this is the way to do it, like to know deeper.
Yeah,
thank you very much.
Thank you for the nice talk. I also see here the problem between cause and effect, and the correlational approach that you have to do, I think, in some cases, just because of the size of the mosquitoes, in the way that when you're measuring one guy, that's done. I mean, there's nothing to do, after that. I don't know how to solve this. I don't know if, for example, if you find that you have a higher diversity of the microbiome in mosquitoes that are infected, and then somehow you can play with the level of diversity in the microbiome in other mosquitoes and see if they are infecting more or less, or I don't know if you think that there is a link between oxidative stress and the microbiota, and then you can also expose to some substances that are going to alleviate or to induce oxidative stress, and this is going to cause a consequence on the microbiota. I don't know if you are planning something like that.
Yeah, indeed, the next step for the next years, we are going to assess these kind of interactions, but varying the temperature of the mosquitoes to see, for example, how it can be affected, and of course, this is the next step to assess the effect of the temperature, and in the future we will see. Of course, we have to go deeper, but trying different environments. But it's really difficult to control the microbiota diversity of the mosquitoes if you don't go from the beginning, removing all the microbiota and exposing them to a specific bacteria you want to assess. It's really difficult because in the field, it depends. For example, in this case, these mosquitoes were collected during the larval stage and a specific water body, and you can see that Faecalibacterium, some bacteria that they could be involved, for example, or related to the feces, the human feces. But if you collect these mosquitoes, the larvae of Culex pipiens of the same species, in other water body that is cleaner, maybe you are going to find a total different microbiota community. But in both cases, for example, we can see that Wolbachia is there in Culex mosquitoes, in Culex pipiens, for example, and not in Culex perexivus. So it seems that the species has something to do, but it's not everything. The environment is really important.
You said these were larvae, right? And that they're getting it from the water. Did you also sample the water in those cases?
No. Not in this case.
Okay, we have one more question online. How much variation in microbiota composition exists between individuals within the same group? So, for example, controls or treatment.
Sorry, can you repeat?
Sure. How much variation in microbiota composition exists between individuals within the same group? So, for example, within the controls or within the treatments.
Okay, it depends. Normally, it's not too high. There is an individual variation, but the dominating bacteria used to be the same, and it's not a big variation. We can see it, for example, here in the field. There are variations, of course, because the different individuals have different habits, for example, or different behavior. But the most dominant bacteria, it's are maintained normally. So I hope this answered the question.
There are some comments from the
Okay.
So there are some comments from the for the first question from the online. Daniel Sanchez, he said, "I understood they were correlated to the infection state."
Yeah, this too. Yeah, but this result is from a study in which the authors exposed the mosquitoes to West Nile virus, and they observed that infected individuals presented a higher relative abundance of Serratia and Enterobacter. And if he refers to the bioinformatic analysis, they compared the total microbiota composition using 16S ribosomal RNA gene as well. So
Okay.
I don't know if
he also mentioned that he has some results of higher microbiota species richness in affected ants, but he's still struggling in deciding what is the correct interpretation of it. How do you explain the increase?
How I explain the increase? Yeah, I will expect, because I didn't say that, but in this study I remember that they also observed an increase in the an upregulation of the immune response. So how I explain that is that mosquito microbiota is usually dominated by one or two taxa, normally one taxa. So this upregulation of the immune response can affect the population of this dominant taxa, and and if this population decreased, it led other taxa to increase in the relative abundance. So this is our hypothesis so far.
Okay, thank you.
Okay. There are no more questions. I think we're done. Very well done, Marta.
Thank you.
And see you all next week, right?
Yes.